Hi, welcome back. This is Ronnie Powers, founder and CEO of Pluto. This video will be a quick follow-up to our last video on differential expression analysis in Pluto. Check out that video for more information, but you can see that the outcome of our differential expression analysis were these two different volcano plots that are ready to go in Pluto, and we've customized one of these plots to show off some of those options as well. Now, a common follow-up after performing differential expression analysis, which tells you about the genome-wide gene expression changes between two different groups, would be to perform a type of pathway analysis to tell us what kinds of biological pathways and processes are these genes that are changing expression involved in. That tells us something biologically meaningful about the condition that we're studying. In this particular experiment, we're looking at gene expression in SARS-CoV-2 positive cases versus healthy controls. So ideally, we're trying to find some inflammatory gene expression profiles or things that are consistently changed in the COVID-2 positive patients. I clicked this Add Analysis button to open up our analysis pane in Pluto, where you'll see that I can select from a variety of different analyses, including a pathway analysis called gene set enrichment analysis. In order to perform gene set enrichment analysis, you need to have already performed a differential expression analysis. You'll notice that if you haven't yet performed differential expression analysis, this gene set enrichment button will be disabled. To perform gene set enrichment analysis, I'll select this button and click Next, and we'll set some parameters for our pathway analysis. First, you'll see that I have two different differential comparisons, one for all COVID-2 versus control and one for just the female samples. I can select which of those comparisons I want to perform my pathway analysis on. I'll select just the female comparison in this case. The second step is to select a gene set collection. These are collections of previously annotated gene sets from the MSigDB. A common first one to take a look at is this hallmarks gene set collection. Click Run to kick off a gene set enrichment analysis. You'll see that initially that analysis returns what's called an enrichment plot. This enrichment plot is pretty typical for gene set enrichment analysis. You might have seen them before in publications. Really what we're looking at here is our ranked list of genes according to fold change in this case. And each black tick mark on this bottom axis represents a gene that was annotated in this hallmark interferon gamma response pathway. The plot also restates the comparison that we were looking at. And the enrichment plot that we're looking at here is for the top enriched gene set by p-value. If you've run gene set enrichment analysis before, you're probably aware that there are lots of other different gene sets in this collection that we chose, the hallmarks collection. So we may be interested in digging in more. On the plot tab, you'll see that for gene set enrichment analysis results, I have different view options. I'll stick with an enrichment plot for now to show you how you can edit the plot title or subtitle. And you can also select a different gene set instead of the one that I'm showing here, which is the most significant. To do that, you can expand this table to view all gene sets that were in the collection, sorted by their adjusted p-value, and take a look at those normalized enrichment scores. A normalized enrichment score that's a positive value represents a pathway that was upregulated or consistently increased those genes in your experimental condition, and a negative normalized enrichment score means that the genes in that gene set were downregulated pretty consistently. Using the drop-down menu here, I could select a different pathway, for example, this oxidative phosphorylation pathway, and click View Changes to update the enrichment plot that I'm looking at. This will take just a second to update, and we'll see that oxidative phosphorylation was one of those negatively enriched gene sets. This is another great use for the clone feature in Pluto, which would allow you to view multiple enrichment plots in the same experiment. We can also view multiple gene sets on the same bar plot, choosing this score bar plot. So here I can select a p-value threshold, maybe make that less stringent, and I want to show both negatively and positively enriched gene sets, and click View Changes. Now we'll see a score bar plot pop in, and we should make that full width so that we can appreciate the aspects of the plot more. Now what we're looking at is a ranked list of all of those different gene sets that met our p-value threshold here. As with other analyses in Pluto, you can view the methods including the R packages that were used for this particular comparison, and easily copy and paste those methods into a publication or presentation that you're working on. Additionally, the underlying data for this plot can now be seen on the Results tab. Where we were looking at before, our DEG tables can be selected, where you can also see this top enriched gene sets table. So this has all of the data that's shown on that bar plot above. This can be downloaded and used for your own analysis. That was a quick overview of running gene set enrichment analysis in Pluto. Lots more customization options and everything to explore, but I hope this was a helpful overview, and we'll see you in the next one. Thank you.